Hi everyone, in this video we are going to discuss about secondary growth part 2. Key points. If the heartwood is destroyed in any stem, then there will be no effect on plants. Any vital function is not affected. But if the sapwood is destroyed, then the plant will die because conduction of water will be blocked. Study of wood is known as xylotomy. If a wood is exposed freely in air, then decomposition of sapwood takes place rapidly. Position of oldest layer of secondary phloem is just inside the primary phloem. Position of youngest layer of secondary phloem is just outside the vascular cambium. If we remove the hardwood of a stem, it cannot affect that plant. Why? Because it is economically important, but there is no effect on biologically. But sapwood is biologically important, particularly physiologically important in water consumption. If we remove sapwood of a plant, then plant will be die because of conduction of water will be blocked. The study of wood is nothing but xylotomy. The oldest secondary phloem is just inside the primary phloem. The youngest layer of secondary phloem is just outside the vascular cambium. Position of oldest layer of secondary xylem is just outside the primary xylem. Position of youngest layer of secondary xylem is just inside the vascular cambium. If xylem is blocked, then shoot will die first. See, now coming to xylem, the old xylem or secondary xylem is just outside the primary xylem. The position of youngest xylem is just inside the vascular cambium. If xylem is blocked, then shoot will die first. These key points are very important in the exam point of view. Check these points carefully. We already discussed about activity of fusiform initials. Now the activity of ray initials. Periclinical divisions are also going in continuously in ray initials of vascular cambium. Some of the cells are formed towards inside and some of the cells are formed towards outside through these divisions, and these cells are made up of parenchyma. Radial lines of cells of parenchyma are formed in the stem. These are called vascular rays or secondary medullary rays. They conduct water and food in radial direction. Order of development of vascular rays is both centripetal and centrifugal manner. They pass through secondary xylem to secondary phloem in radial manner. Formation of annual rings. Springwood and autumnwood formation. Annual rings are formed due to unequal activity of vascular cambium. The activity of cambium does not remain same. It is changeable throughout the whole year. Activity of vascular cambium is affected by physiological and environmental factors. In winter or autumn season, the activity of the cambium is less and forms fewer xylary elements. The cells formed during this period are small, thick-walled, darker, with higher density and have narrow lumens. This type of wood is called autumn wood or late wood. The vascular cambium is highly active in spring or summer season and produces a large number of xylary elements, and cells of secondary xylem are larger, thin-walled, lighter in color, with a lower density and have wider lumens. This wood is known as spring wood or early wood. The numbers of annual rings formed in a tree give the idea of the age of tree. Study of determination of age of plant called dendrochronology. The annual rings are counted from the base of the stem because basal part has maximum annual rings, upper part has lower annual rings. Therefore, counting from the basal region can give the correct idea. The width of annual ring is less in unfavorable conditions and more in favorable conditions. Thus, the annual rings are also indicative of environment period the tree has encountered previously. The autumn and spring wood are formed in the form of rings. The rings of any type of wood is called growth ring. Thus, two growth rings are formed in 1 year and they collectively constitute a annual ring. A piece is taken from the stem, from the base of the stem with the help of increment borer instrument. The annual rings are counted from that piece and again inserted or fitted into the same stem at the same place. More distinct annual rings are formed in that region where climatic variations are sharp. Distinct annual rings are not formed in tropical plants. More clear annual rings are formed in deciduous plants as compared to evergreen plants, particularly in temperate region. Least distinct annual rings are formed in seashore regions because climate remains same throughout the year. In deserts, annual rings are less distinct. Distinct annual rings are not formed in India except Himalayan regions. More distinct annual rings are formed in temperate plants. The study of age of a tree is called dendrochronology. The study of age of plant was done by its annual rings. Annual rings are formed by spring wood and autumn wood in an year. All these points are very important in exam point of view. Check these points carefully. Up to here, steel or secondary growth in dicot stem was completed. In next video class, I will explain you about extra steel or secondary growth in dicot stem. Thank you.